My name is Francisco José Sánchez Luque, and I am a tenure scientist, a principal investigator in the Institute of Parasitology and Biomedicine López Neira from the Spanish National Research Council, and I am the group leader of the group of molecular genetics of mobile and foreign DNA. The focus of our research is in the particular dynamic of mobile DNA and integrative viruses like HIV. So these subgenomic entities share the possibility to invade and propagate copies in our genome. They not only participate in viral infections, but they also, when they're regulated, can contribute to neurodegeneration, cancer, and senescence. All the genetic sequences that populate our genome, they have acquired regulatory mechanisms, both either targeting the DNA or also the RNA intermediates. Most of these mechanisms we are now learning how they occur, but there is still a lot of them that remain unknown. And the goal of my laboratory is to elucidate this kind of regulations, the pathway they share, and how we can intervene for improving health conditions or treating diseases that are related to this kind of sequences. In the future, our major interest is to develop the knowledge that we are accumulating now about the regulation and the ways to target these sequences, to develop into therapeutic tools or other technologies that could be used to prevent the deleterious activity of these elements and facilitate technologies or therapies like, for example, regenerative medicine or direct health conditions like aging, neurodegeneration, or other sort of neurological disorders that are known to have a contribution through the regulation of these elements. So maybe the most recent research output that I have had in my career was the characterization of one of the regulatory motifs or regulatory sequences that are used by mobile DNA and probably shared also with other retroviruses like HIV to acquire a regulation by our cells. This was sort of revolutionary because typically these elements have been seen as selfish DNA trying to escape from our control, but my results show the opposite, show that they have acquired along evolution a regulation probably to modulate their activity to tolerable levels for our species. So, as I said, we are now learning a lot about the contribution of this component of our own genome, this mobile DNA that populates our genome, into many health conditions or diseases that humans experience, either by triggering or by contribution to progression. One of these cases is very dramatic, is in certain type of cancers. Understanding the mechanism to how our genome regulates the activity of these sequences can be essential for in the future develop more accurate therapies or approaches to control and minimize the effect, the impact that these sequences have in our health.